So next up, Radek Erban from Oxford is going to talk to us about some recent work connecting molecular and Brownian.
Oh, thank you very much. So in January we had opening workshop of this research program, and what we did, we discussed many open problems in the research area, which were also summarized this morning during the program overview. And we also started with a talk which is called 8 Open Problems, when we try to kind of formulate some problems in their area. So the first problem which we discussed was this open question 1. Can we simulate complex intracellular processes which take place across the whole living cell with fine atomistic resolution of molecular dynamics? So in my talk I would like to kind of summarize the progress which we have made on answering this question during the last 6 months. There can be obviously many reasons to answer this question negatively, just when you think what actually all atom molecular dynamics is. So here I then little illustrative snapshot from a simulation which I'll be discussing later, which is a simulation of ions in water. And what we do in our molecular dynamics, we describe all atoms in the system, their positions and velocities, and store them in long vectors x and v, and then we just solve Newton equations. So the resulting model is just a nonlinear systems of all these when these are very long vectors, which is to be discretized with some time step. Typical time step of molecular dynamics simulation is about 1 femtoseconds. And we have to obviously, for intracellular processes, we are studying biological applications, so it's always MD simulations in water. In fact, I said this is simulations of ions, but you can actually cannot see any ions here, just water molecules in this snapshot. So MD simulations are limited to very small domains containing only several thousands of water molecules, while the whole cell would contain like 10 to the power 10 or 10 to the 12 water molecules. So from just these points you can conclude that there are many reasons why we could just say no, and my talk could finish just in 1 minute, because we have problems just to store all this information in a computer. If you are able to store it, then you not advance the system over a reasonable time scale. But we spent here 6 months, and I would like to kind of, and we are kind of not optimists, so I would like to show you that there is some hope to actually positively answer this question. And that's what I'll be doing in my talk. So as I said, it's impossible to use a computational intensive MD model over the whole simulation domain, but in some applications, microscopic detail is only required or known in relatively small regions. So there's a potential to positively answer this question by developing suitable multi-scale hybrid methods, which use models with different levels of detail in different parts of the computational domain. So in this talk, I will discuss 4 methodologies for multi-scale hybrid modeling. And in order to orient in my talk, I'm going to draw a diagram which will keep appearing in my talk again and again. So in this diagram, I have put 5 different modeling approaches. In the center, I put particle-based stochastic reaction diffusion models, or Brownian dynamics. That's the models which we already seen, for example, in the talk by Maria in the morning. And we will see them in many other talks. On the right, I put more coarser models, like models which are less detailed, like compartment-based reaction diffusion master equation or even mean field PDEs. And on the left-hand side, I put much more detailed model, including molecular dynamics. Kind of motivates this talk. So what I'll be interested mostly in these arrows, how we can derive one model, coarser model, from a more detailed model, and then how to exploit this knowledge, these limiting processes, to design accurate and efficient multi-scale methods. So that you use a hybrid scheme which use models with different levels of detail in different parts of the computational domain. I'll start with this red arrow, which is kind of our biggest success story. It's a joint work with many people, many program participants. I would call it a success story because we have in this area we started with papers on the development of new multi-scale methods. We also now have papers about arrow analysis of these methods, so we actually know how accurate are the computed results. We also have some applications where we are able to answer questions with a level of detail which was not possible before by applying these multi-scale methods, and it's also a software implementation now in Smalldyne, widely used software package. So this is kind of a success story which kind of be great to replicate in this level molecular dynamics or Brownian dynamics, but here I can only present some work in progress, so something which we've done during the last 6 months, also various things which could be done in the future. There are already some partial results published in these papers, and I'll also discuss some unpublished work. Okay, so I said that I'll put to the center this Brownian dynamics, so I should maybe explain what I mean by Brownian dynamics. So in the simplest description, Brownian dynamics, it would be like a protein or Brownian particle, and its position at time t, and I compute its position at time t plus delta t by adding some random displacements, which is normally distributed. So if I iterate these schemes over several time steps, I get typical behavior of Brownian motion, this Brownian path, which will be just a discretized equation, discretized SD in this form. So this will be just in terms of trajectory Brownian dynamics, but also I have to describe some events. So just in reaction diffusion processes. So reactions in these models are described often in terms of some binding probability, reaction radius. If I have two Brownian particles, if they are sufficiently close in some reaction radius, then they can react. So a long time ago, we studied these schemes with John Chapman, but there's plenty of work in this research area. It's a very active research area, and there were papers coming from this program studying these models. There was also a lot of work recently on software implementation. Now, many software packages to choose from. I'm not saying they are the same, but they are kind of underlying the same philosophy. They're simulating individual proteins, individual particles. So I would like to say that in my talk, I will consider this Brownian dynamics term slightly broadly. So I'll also include in later parts in this group models which will be written in terms of Langevin dynamics. So for my Brownian particle, I not only describe it by its position, but also by its velocity, which is subject to friction force and some random changes. I'll also discuss models which are relevant to polymer modeling, which is a DNA modeling. So it's kind of the coarsest DNA model, which would be in terms of bead spring models. So I have several beads connected by springs. So again, these beads are subject to Brownian motion, but now there are also some spring forces. So this was an introduction of what I mean by Brownian dynamics in this talk. And now I would like to discuss this first multi-scale methodology. I would like to take a Brownian dynamics simulator and replace it in parts of the domain by coarse approach. And I would like to get the same results by my multi-scale model as I would get by Brownian dynamics. That's kind of the philosophy. So in order to explain that, to tell you what actually is this compartment-based approach, so I will explain what is this Brownian dynamics. So I'll start with the simplest possible, like discretized Brownian motion. And I can formulate the compartment-based approach in a very similar way if I was thinking what actually individual particles are doing. So if I would formulate it as a jump process on a lattice. So I have a lattice point distance h apart, so this is my compartment size. I kind of put these particles in the middle of these compartments, and they can jump in all 6 directions, or they can stay where they are. So if so this will be the kind of evolution rule, which is kind of equivalent to this evolution rule when I calculate position of the particle on the lattice at time t plus delta t using position at time t by just adding h into different directions, different probabilities. So if I do it with these probabilities in correct limit, I get again diffusion process. So there will be no advantage using this diffusion process over this diffusion process, because kind of if I was studying the behavior of individual particles. But the big advantage of compartment-based modeling is that in practice we actually don't simulate jumping on a lattice. We actually disguise the system by numbers of molecules in different compartments. So the state of the system is just have 7 molecules here, 8 molecules here, 4 molecules here, and so on. And we only simulate the time evolution of the numbers of molecules in each compartment, so which leads to discrete space continuous time Markov chain or reaction diffusion master equation. Again, it was discussed and it will be discussed many times during the workshop. And then plenty of people, including people in the audience, who work on this on understanding properties of reaction diffusion master equation, and also I saw some nice posters. So having introduced these approaches, I would like now to discuss hybrid modeling. So it's kind of a nice area to work for a mathematician because we can find well-defined mathematical problems here. It's kind of well-defined in a sense that, so assume that this is my domain, and here I just will use Brownian dynamics in this part of the domain and compartment-based model here. What I would like to achieve by my multiscale approach, I would like to achieve the same spatio-temporal statistics in this subdomain as I would get if I was using Brownian dynamics everywhere. So that's kind of a goal. It's useful in application. I will mention some applications later. In applications, the picture is slightly different. So obviously, this more detailed domain is much smaller. So it will be kind of very small, and the rest of the cell I will just use compartments. And there are many different solutions. The obvious solution, which on the face of it looks like the easiest solution, is just to try to simulate everything using the approaches which I just described, and somehow kind of use the same rules for transitions according to this artificial boundary. Because I know in compartment base I'm just jumping on the lattice sides with some rate which is the diffusion constant divided by the length of the compartment size square, so it's a d over h squared in my notation. So I also know what would be the jump rate from here to here if there was a compartment. It would be just d over h squared. So I could simulate this random walk, and when I'm here there will be some probability that I will jump to the right. I could then initialize the particle here. Again, I could think there's some analogy. Maybe I should initialize it like a half compartment, like put it here, the particle. Then I could do Brownian motion, and if the particle wanders here, then if there are 7 particles, I will change my state in this box to 8. So if you do it this way, then you obtain significant errors. So it can be seen already when we simulated just a diffusion at equilibrium. So I will not be simulating any reactions, which is considered diffusion at equilibrium, and I put particles in a box, and I would like to just get a homogeneous profile. And this is the result which you would obtain. So it's a diffusion, so I would like to get, this is my average concentration, so I would like just to get a constant. But you can see that in this simulation, when I basically use the compartment-based model here and Brownian dynamics here, so what I'm plotting here is this is just numbers of molecules, different compartments. Here I have many particles, so what I have to do, I have to, at the end, discretize my domain and calculate how many particles are in each compartment, so I could visualize it in the same way. You can see that in each of these subdomains, I get a diffusion, I just get a constant concentration, but there is significant error here. There is some boundary layer, some boundary layer behavior on this artificial boundary. And the goal of these multiscale methods is obviously design them in a way that nobody will see this boundary, because this is just a modeling choice. So why such a naive coupling gives such large errors? Well, it gives because although these approaches are kind of both okay to simulate diffusion process, and they give the same statistics of diffusion process, they look quite different at a microscopic level. So if I think about what is happening in this Brownian dynamics part, so so this is now time, this is space, so this is my Brownian dynamics domain, this will be already first compartment. So if I'm position xt here, then I just use this update rule to calculate the position at time t plus delta t by adding this random displacement. So I just calculated the particle is here. But the particle doesn't go straight. If I actually refine the time step, then I would find out that in some trajectories actually are crossing the interface, so maybe I should have, in my scheme I would just, with a smaller time step, transfer this particle here, but some particles not. So you can see that the rate of transfer across this boundary will depend not only on the experimental measurable quantity d, but also on the internal parameters of each method, like for example time step delta t here. So in order to design correct coupling which would not see this boundary, we have kind of design coupling which would also depend on internal parameters of both molecule-based and compartment-based models. So in this case, it would be just time step delta t of the molecule-based model and compartment size h. There are many ways how to do this, so I'll just show you one approach which works. So this is just I put 3 rules on my boundary, so I'm just basically doing the same thing, but I'm slightly changing the rules on the boundary. Before I told you that particles which are jumping from this boundary compartment with the standard rate, which is d over h squared, but I now say they will jump with some d over h squared multiplied by some constant. So if phi is 1, I get the previous model. Then I have to somehow decide, this is a much more detailed model, so I have to kind of decide where to initialize particle, because here I only know that it was in a compartment. So I just say that a particle which jumps here from here, it's initialized at a distance x from the boundary, where x is sampled from some probability distribution. And all particles which wander here are kind of accepted to the boundary compartment. So now the question is, how do we relate these coupling parameters, which I introduced, phi and fx, with experimental data, which we diffusion constant d in this case, and algorithm parameters, which is compartment size h and time step delta t. So to do it, we have to kind of write some governing equation for this coupled process. So this I just put them here. So this will be an equation for the probability of a particle to find in the last compartment. So I denote this compartment K. So it's a probability to find particle in the K compartment. This is just coming from the compartment-based jumping. Just this lattice-based jumping is a standard master equation. And this is the other part, which comes for the particles which were in the Brownian dynamics domain, and they are all transferred across the interface. And in the Brownian dynamics domain, I describe particles in terms of probability distribution function. They are in continuous space x. The particles are diffusing, but also some source term, which there are particles which are introduced according to this rate, and they are placed at the position fx. So now I have this type of equations, and I want to kind of find the appropriate values of phi and fx. So what are the conditions which I want to place on them? So, well, I already know that if I choose phi is equal 1 and f will be just a Dirac distribution at h half, then I get very significant errors. I get some boundary layers. So I would like to find phi and fx in a way that there are actually no boundary layers, there are no errors on this artificial boundary. So I can do some asymptotics, which is kind of published a few years ago in this paper, and I obtain the following formulas. So these formulas are just for phi. So phi is not 1, but it depends on h and delta t. It could be, depending on what is h and delta t, could be either greater than 1 or less than 1. So I either accelerate the rate of particles or decrease it. And this is the distribution. Now, instead of showing you the derivation, I'll just show you just like illustrative movie. Just shows that these formulas work not only at equilibrium, but also work in out of equilibrium simulations. So in this simulation you just see a very simple test problem. Basically, I put all particles that have a domain. All particles I put on one side of this domain, and they are just diffusing. So at the end, the equilibrium will be again just constant concentration profile, and it's kind of in a loop, so that's why it's jumping. So what I'm simulating in this part of the domain are just 10 compartments, and here I'm simulating individual particles. What you see on the right is the visualization of the same process, but I just bin this subdomain, and I put and I have put just how many particles are in each subdomain to get this profile. And what I'm plotting here as a blue curve, this is the correct solution. This is just a diffusion, so I can exactly calculate some average profile in this case. So obviously you cannot see like much you can see as a noisy, but I can easily, in this example, increase number of particles and just show you that there is no observable error on this boundary. So then that would be the same simulation, and just increase number of particles. And you can see that there is no error or visible error, or no boundary layer visible on this artificial boundary between different domains. And I'm starting out of equilibrium. Obviously, in equilibrium it will also work. So it's kind of like illustrating that with some tweaks we can design a scheme which will not have this like a boundary behavior, which will also give me correct spatio-temporal statistics in this Brownian dynamics domain as I wanted. So, but there are obviously many different solutions. So I discussed just this two-regime method developed by Mark. Later, we are discussing several other approaches, and one of them, which I'm presenting here, is adaptive two-regime methods, which kind of is useful for problems with moving interface, because here I kind of postulated where is my boundary between different regimes, in some problems, like for example, with problems with traveling fronts, this is the boundary between different regimes. This is the boundary between regimes when I need when I have only a few particles and a lot of particles changes. So again, I will not show. You can see find the method and the analysis here. I'll just show the movie just to illustrate what this method is capable. So in this movie, I'm just illustrating it on a reaction diffusion traveling front, just a Fisher equation. So here we have I'm comparing 3 different simulations. One of them is a PD, so it's just a Fisher equation PD. So there's some traveling front which is traveling from left to right. What I'm also showing here is the corresponding compartment base model. So I'm using the same reaction scheme, and you can see that the compartment base model is quicker than the PDE for this particular parameter set. I can also do the Brownian dynamics very detailed when I consider individual particles, and you can see the Brownian dynamics is slower. But what the of this picture is this first picture when I use this Brownian dynamics, this complicated computation intensive part of the simulations only in the front of the traveling wave when I have only a few molecules, and the rest is just replaced by compartments. And I'm getting the same behavior, same traveling speed by this multiscale approach as I would get if I use Brownian dynamics everywhere, as you can see in this video. So that's another like a type of multiscale approaches we have been working on in this area of coupling Brownian dynamics and compartment-based model. There are several other methods introduced by, there are several other methods introduced by Mark Fleck, and we also a year ago we got software implementation for this 2-regime method in SmallDyn. So now it's part of SmallDyn. SmallDyn was in a particle-based simulator. Now it's been extended to a compartment-based simulator as well, and there is also multiscale method included in SmallDyn. Steve Andrews, who is a developer of SmallDyn, also was a participant of this program during last 6 months. And I promised some applications. I'll just briefly mention them if you are interested in applications. So we applied this was work of Ulin Dobramester to modeling calcium dynamics, modeling calcium puffs. So in this, in this situation, we are modeling calcium-induced calcium release from endoplasmatic reticulum, and most interesting dynamics is happening close to receptor. There are some binding sites when calcium can bind, and then the channels will open, and there is a huge influx of calcium. So if you look on the time trace, this is time, and this is concentration of calcium, you get these huge spikes. So it's a very nice problem for multiscale model, because you get times when you have only few calcium molecules around, and then you have spikes when you have many more calcium, and you are able to obtain this time trace by exactly using this multiscale approach. That close in this small subdomain we were using Brownian dynamics, but the rest of the domain, most of the cell was replaced just by compartments, and we just use this multiscale approach. Another application area does work with again with Uli, another program participant. Garrick Papoyan was working on actin dynamics in Philopodia. So Philopodia are small protrusions from cell membrane which cells use to export an environment. They are made from actin filament actin bundles which grow. So there are plenty of processes when the actin binds. This makes filaments longer. Also some proteins, capping protein stops polymerization. There's also depolymerization. There's also retrograde flow which pulls filaments back. So it's a nice dynamic system, but again a lot of interesting things are just happening close to the top. So we can use detail approach here, and using compartments which kind of highlighted here in the rest of Philopodia again to achieve kind of Brownian dynamics resolution with less effort than you would get if you were using Brownian dynamics everywhere. Okay, so this was kind of like an overview of this first arrow. Now I can show you not just our result, but something which we kind of wanted, we still would like to replicate in these other areas, but here we only have some like a partial result, so always work in progress. So the first thing which I can just mention is that you can obviously couple Brownian dynamics if it's the model which you want to write your or method which you want to write your models in. You can couple it by other Korsgen approaches, and one of them will be just to couple it with actually a mean field partial differential equation. So if I have this setup, the mathematical problem now would be that I use Brownian dynamics here, and here instead of compartments I will be just using reaction diffusion PDEs. So it's quite a difficult problem. We wrote one paper about it 2 years ago. It's difficult, and it's kind of easy to explain why it's difficult, because it's a fundamentally different problem from the problem which we are facing here. Because here we were coupling one realization of stochastic process with another realization of coarser stochastic process here, while here we are coupling one realization of stochastic process with mean field equation, with the equation for average behavior. Nevertheless, you can try to design some schemes, and you can actually play just with diffusion in this case. So one could design various schemes, and which are in this paper, which basically mean that I'm solving concentration profiles here. I'm simulating particle dynamics here. Whenever particles cross, then they are introduced as Diracs to these concentration profiles. What we found that for all schemes which we could kind of come up with, we could get correct average behavior for this process, but we couldn't get correct fluctuation here. So the only solution is that one actually is to introduce some overlap regions, which is very common in multi-scale applications. It's a region when these different descriptions exist together. So you would solve a diffusion PDE just on this domain, and you would kind of initialize particle and on this part, but you let them diffuse all the way until here, and then you can get correct variances. The disadvantage of any overlap region in this research area is that you can, if you go beyond diffusion and you want to study some more interesting problems, when there are some reactions, you have to start to think how you model reactions in these overlap regions, how the field would react with particles. So that's and this is kind of there's more open question than answers in our paper. Anyway, we can get back to this arrow, which I was discussing, studying molecular dynamics, and how we can try to apply the same philosophy on designing multi-scale methods between molecular dynamics and these particle-based approaches. So first, let me remind you what I actually mean by particle-based approaches. So I said in the first half of my talk, it was all about discretization, just Brownian motion. Now I will also include some stochastic-based, particle-based stochastic reaction diffusion models which can be derived from more detailed models, and include Langevin dynamics. So each particle will be described by its position velocity, but also more complicated models where each particle will be described by its position velocity and some auxiliary variables. And also discuss some work which we done in the institute on modeling DNA or polymer chains when these Brownian dynamics particles are also coupled by springs. So the goal is was obviously to answer this question: can we simulate complex interstellar processes which takes place across the whole living cell with fine atomistic resolution of molecular dynamics, or which could be formulated for this community, or can we couple these all-atom MD simulators with particle-based stochastic reaction diffusion methods which have been developed by many members in the audience, and in order to kind of get some understanding what can be done, I'll just start with a simple example. So I will discuss this application about calcium dynamics, where we use Brownian dynamics model to understand calcium puffs. So I will also use as my example just ion dynamics, to study diffusion of ions in water. So if I want to write a very basic, very simple molecular dynamics simulator, so I have to decide how I describe water. There are about maybe 20 water models, so I was kind of design the standard simple model called SPCE. But there are many others to choose from, and I'll put a lot of water molecules into a box, and I put also an ion there. So this will be kind of what is kind of my initial condition, just to visualize it. So it's a box about 500 molecules of water, in which in the middle occasionally you can see that one ion which is hiding there. So now, in order to simulate, design molecular dynamics code, I have to just take all the position and all the velocities of all atoms, water molecules, and the ion, and also take the appropriate force fields and simulate these Newton equations. I have to choose what numerical integrators and should I do to satisfy constraints. But if I do it, then the dynamics would more or less look like this. So in this video, I kind of visualize it by decreasing little size of water molecules and increasing the size of ions, as you can actually see it. And what you can actually see that this ion is more or less doing just some is subject to some stochastic, like a Brownian motion or some like stochastic particle-based approach. So if I remove all the water molecules, and if I knew actually what stochastic process this ion is doing, then I could kind of exploit this understanding of this limiting process to design some multi-scale models which could simulate ions with water in part of the domain and just using some stochastic model elsewhere. So in order to get some insights into this problem, I'll start first with a very simple toy model, which kind of has the similar flavor like this, like all atom MD simulations. So it's just kind of a very simple molecular dynamics model, but I would still call it molecular dynamics. So here in this model, I have a few heavy particles which have mass m and a radius r, and they are in the heat bath of a large number of light point particles with masses much smaller than m. So what is happening? These light particles are just going straight, and they are colliding with these big particles, and the post-collision velocities can be computed and using the conservation of momentum and energy. And what I so if I showed you a video, it would look very similar to the video which I showed you before, in a way that these big particles will be just doing some like a random walks. Here I just put like a snapshot where I put a few big particles, and I showed you the trajectories of these big particles, which are subject to this simple coarse-grained molecular dynamics. Now, what is nice for this model has been studied in mathematical literature, and you can actually prove some theorems about this model. So it was done just studying a paper in 80s. So you can look at the limit m over m, which is the limit of this mass of the small particle divided by mass of the large particle. And if you suitably scale the density of these particles, take more and more particles, light particles, and the initial velocity distribution, then you can actually prove that these heavy particles in this limit will follow the Langevin dynamics written in this form. Not only you will also be able to derive some explicit formula for the friction and for the diffusion constant. So there will be like known functions of MD parameters. So what I can do next, I can actually take this like Langevin dynamics, and in a suitable limit. It's also described for large times by Brownian dynamics. So and I can get it that the Langevin dynamics gives a stochastic model of Brownian motion with a diffusivity which depends on the density of particles, the radius of the big particle, and some moments of this velocity distribution. So there's analytical results. So if I now want to design a multi-scale approach, you can kind of ask what questions I can use these results or adapt them to design the multi-scale approach. So what I would like in part of the domain use this model, and part of the domain use this model. So the main question would be here the particle which is crossing an interface, that this particle is hit by small particles from one side, but it's not hit by small particles from the other side. So I have to kind of compensate by adding some stochastic differential equation to the movement of particles with coefficients alpha I and beta I, which depends on the position and velocity of this particle crossing the interface. But then one can again generalize these results and obtain an analytical formula for these, which can then be used to design multi-scale scheme which doesn't bias movement of particles across this boundary. So particles are crossing this boundary as they would behave on the boundary in the same way as they behave inside the Brownian dynamics domain. In this paper also did just try to apply it to a very coarse model of protein binding. When close to the membrane, I'm using just this type of molecular dynamics, and away I'm using Brownian dynamics. But you can already see that there are some overlap regions. So here on this boundary, I'm using this coupling of molecular dynamics with Langevin. But to couple Langevin and Brownian, I just use some overlap domain when I'm using Langevin dynamics all the way up to here and Brownian dynamics up to here. So it's kind of a toy problem to get some insight into this problem of coupling all atom molecular dynamics with Brownian dynamics. So let me look at what could be done here, and then just kind of try to follow the same philosophy. So I have this all atom molecular dynamics model. I would like to just kind of couple it with some stochastic coarse-grained model, which is valid at the time scales of molecular dynamics, and hopefully kind of prove that this stochastic coarse-grained model in a suitable limit with Brownian dynamics. And this is possible for the model which I put here. So now I'm not using Langevin dynamics, but slightly more coarse model that I'm describing in each ion by its position, velocity, and its acceleration and some auxiliary variables. So I have now 4 parameters to fit. So these parameters can be fit from running all atom molecular dynamics simulations, and can be then used to design the multi-scale scheme. Now, this kind of looks a little ad hoc, how I can come up with this model, but actually this model can be shown it's a special case of more complicated models, which is coming from dynamic force matching, which is one can design models with many more variables, with many more free parameters to fit, and then you can, if you have many parameters, you can fit as many characteristics of molecular dynamics as you would like. But for the coupling in this paper, I just use this to simplify like a 4-variable model. And the scheme then looks as the flavor of the previous scheme. So I'm now 3 models. I'm using molecular dynamics and divide everything in 5 subdomains. I'm using molecular dynamics here, this stochastic coarse-grained model in omega 3, and Brownian dynamics in omega 5. And I have 2 overlap regions which are necessary to either prepare molecular dynamics for ions entering, or just to solve the problem that this is simulated with much smaller time step than Brownian dynamics. It's kind of like an illustration that such a scheme is not biasing trajectories of ions. So this is my boundary between molecular dynamics and coarse approach. You can see that if I start on the boundary many trajectories of an ion, I get same number of particles going up or down, so there's no bias on the boundary. And also, if I just look at diffusion, I can also look to mean square displacement and get by this multi-scale approach what one would like to get. We said this was kind of like an initial exploration. There are plenty of mathematical problems which needs to be still solved in this research area. So in all of these problems which we try to explore, it was molecular dynamics simulation which were the most computation intensive parts of multi-scale methods. So if all simulation which I showed you were running this 1 femtosecond time step, if I can increase it 5 times to 5 femtoseconds, then I can speed up all the simulation 5 times. So all approaches which can increase the time step for molecular dynamics simulations need to be considered are very helpful. And there's a very nice recent work by Maine Lencourt and Charles Matthews when they are able, by considering carefully how these numerical methods for these ODs which are used in molecular dynamics are able to increase molecular dynamics time step to 7-8 femtoseconds with very good accuracy. If you actually don't like molecular dynamics, it looks too complicated for you. Actually, I think there is a lot of mathematical questions in this part of the picture, and try to couple the stochastic coarse-grained model with Brownian dynamics, which is the reason why we want to couple Brownian dynamics, because this is the model used in small dyno or in software packages which are used in this area. So and has to kind of understand how to design these overlap regions and in an efficient way, how to study the errors of these schemes, understanding connections between stochastic congruence model and Brownian dynamics. You can think already about Langevin dynamics, that it offers much more possibilities how to describe reactions. For reactions, we have just these like a binding probabilities, reaction radius, but we could also introduce some potential here. Then when particles are close, then they are subject to some potential, and that's how the chemical reaction happens. So this is we recently explored, well, some nice extent did all the work. So how to actually generalize these results to the case, how to derive Robin boundary conditions for the case when actually the reactivity is not described by some binding probability, but actually by some interaction potential, which is applicable to both Langevin and Brownian dynamics. So that was kind of outlook, what type of questions are here. There is another, I still have, I think, 3 minutes. So I would like to mention last project, which we made significant progress during the program, which is again following the same philosophy, and this is about the multi-scale modeling of DNA dynamics. So in DNA, it's a long, like a polymer chain. You can find many different models to model it, starting from all atom molecular dynamics, which you can then start to coarse grain. This is a nice recent paper about coarse graining all atom molecular dynamics by changing, using coarser models further from the chain. And on the coarsest level, it could be described by bead spring model. So when this is unpublished work, we are studying how we can actually do multiscaling, not just by replacing, say, waters outside of the DNA by some coarser particles, but also coarse graining along the chain, which naturally leads to a multi-resolution bead spring model. So it's a generalization with standard bead spring model by considering beads of different sizes and springs of different spring constants. I just show you a video. It's a very just video from this weekend, which Edward, I should mention this is the work of Edward Rolls. This is just showing a very simple test problem for this type of multi-scale approaches. This is just a bead spring model of polymerizes, let's say a caricature of a protein transcription factor trying to approach DNA. And what I'll show you as it approaches is that in this method we can take part of the chain and just start to simulate with, introduce many more beads and stiffer springs to just resolve part of the region with much more detailed resolution with the region which is around this transcription factor. So what we did in this work, which is hopefully will be submitted within a month, Edward derived some formulas for diffusion constant and root mean square end-to-end distance of such a model. And since he has a formulas, he can for this multi-resolution model, you can then start with a very fine model of the polymer and then replace part of the chain by coarser models to design a multi-scale approach on both spatial and temporal scales in a way that a coarser region of the polymer chain is then simulated using a larger time step, while you need a very small time step in this region. Anyway, just to summarize my talk, I have discussed these 4 methodologies of hybrid modeling, of using models with different levels of detail in different parts of the computational domain, focused around the success story when we have many answered questions, and I also discuss this work in progress on coupling molecular dynamics with coarser models, sort of Brownian dynamics with PDs when we have more questions than answers, but hopefully within the next few years, as a follow-up of this program, we will make even more progress in this area. And so this was the work done with many people, so to just acknowledge them here, and it's also funded by many funding bodies. And if you are interested in some preprints, you can find them on my website. Thank you very much for your attention.